we present the activities carried out by some of the few hundred scientists involved in the Bioneca project. Among all the interesting contributions, I would like to emphasize the ones related to the total artificial heart production, to the use of ozone therapy to treat the COVID-19, and to the neural stem cells in space and biomaterial fabrication in International Space Station. The main goal of this project is to establish an intensive interaction about communities involved on one side on physics and other basic sciences, and on the other side on regenerative neurology and cardiology, in order to reduce the number of deaths associated to brain and heart diseases. I decided to start to write the Bioneca project when I realized that our idea to track stem cells in cardiological experiment by using the X-ray synchrotron radiation microtomography was successful. Figure A and B shows an infarcted heart of a mouse in which stem cells were injected. Figure C shows the distribution at the three-dimensional level of the stem cells with an accumulation in the lowest part of the heart, namely in the infarcted region, which is zoomed in the figure D. The project is organized on 5 working groups involving scientists belonging to 38 countries.
Heart failure is a major public health problem affecting more than 40 million people in Europe. It is a large chronic epidemic disease with an increase in prevalence related to the aging of the population. New viral infectious disease like COVID-29 induces viral myocarditis in the recovery period, frequently turning to chronic heart failure. Many heart failure patients become refractory to medical therapy, necessitating more active therapies like biotechnology approaches, resulting in the creation of the tissue-engineered bio-artificial myocardium. That's why the cardiopatch and cardiowrap bioprosthesis or biomechanical artificial heart, like the Carmat bioelectric device, are new emerging tools. Cardiovascular tissue engineering involves the creation of myocardial tissue heart valves and blood vessels. This approach represents a new hope for the creation of biomimetic tissues like myocardium and bioartificial organ. Myocardial tissue engineering has been explored with aims to repair, replace, and regenerate the damaged cardiac tissue. The association of stem cells and elastomeric scaffolds in the form of cardiopatch or cardiowrap avoid ventricle dilation and heart failure. This should avoid heart transplantation. Cardiopatch and cardiowrap bioprosthesis are indicated to support myocardial infarction and other myocardial diseases, limiting the spread of the pathological areas and improving heart function by reducing stress by a girdling effect. Carmat Total Artificial Heart is composed of implantable bioprosthesis and a portable external power supply system to which it is connected. Carmat intends to overcome the well-known shortfall in heart transplants for the tens of thousands of people suffering from irreversible end-stage heart failure, the most seriously affected of the 20 million patients with this progressive disease in Europe and in the United States. Carmat is the first physiological artificial heart using highly biocompatible materials. Manufactured with self-regulation and pulsatile systems, the Carmat Total Artificial Hearts are showing successful clinical results. Potentially, Carmat might save lives of thousands of patients each year with no risk of rejection and with a good quality of life.
Good morning. I'm Barbara Zavan from the University of Ferrara in Italy. Ferrara is a small city in the northeast of Italy between Venice and Bologna. Ferrara is well known thanks to her amazing castle inside of the city, and Ferrara is a patrimonio of UNESCO that also the Chirico use and love its main castle. I work on tissue engineering and regenerative medicine, that is the field of biotechnology, which aim is the in vitro, in vivo reconstruction of tissue and organs, with the aim to perform the translation from traditional medicine to personalized medicine. This is my lab, on which I work with several postdocs and several fellowships on study how the cells work and talk with the biomaterials. I study this talking thanks to the morphology, but also by molecular biology analysis and intracellular signaling, mainly for mitochondrial physiology. We work with several types of cells, adult cells, but also mesenchymal stem cells, embryonic stem cells, amniocytes, and so on. We use these cells and several different types of biomaterials and 3D printing in order to obtain and to study how the cells are able to reorganize in vitro and in vivo to perform this in vitro reconstruction of tissue and organs. In my lab, I work with several different kinds of specialty, with clinicians, but also with biotechnology, biology, chemical, engineering. And in last year, my attention was focused mainly on exosomes for this in vitro reconstruction. The exosomes are these small nanoparticles that are produced mainly by all the cells, but for the tissue regeneration for mesenchymal stem cells, because are the first who are involved in tissue regeneration, who contain several signals in order to improve the regeneration or to immunomodulate. These exosomes are produced by the cells. If we change the environment, that could be the epoxia, the scaffold, or inflammation, and we are able to play with stem cells with different kinds of biomaterials in order to obtain different kinds of exosomes with different kinds of activity. We are able to isolate the exosome with several types of technology and to analyze in several ways. We are able to analyze the exosome if they are produced by the cells in monolayer or in 3D culture. We are able to quantify, to analyze in size, but also by their content in the extracellular membrane with the typical CD, by electron microscopy, by both TEM or also SEM. We are able, moreover, to analyze, for example, the activity of the exo when they are put on the cells. For example, here are cardiomyocytes that are treated with the exosome started to induce a calcium intracellular signaling and start to contract. Moreover, we are able to interpret this exosome to several types of biomaterials. This is, for example, an hydroxyapatite, in order to obtain a 3D scaffold enriched with this exosome and able to improve the regeneration. But we are able also to use the hydrogel for soft tissue reconstruction and put everything in 3D printing in order to obtain a personalized specific organs.
Joseph Bohajar, and I will be presenting a little bit the work that we have been doing with relation to the COST project Bioneca. A quick introduction to the University of Malta. It has 400-year-old history. We have 14 faculties, some institutes and some centers, and approximately 3,500 students graduate annually. In the project Bioneca, myself, Joseph Bohajar, and Professor Inge Bertram Mallia are both MC members, and we participate actively within the Bioneca project. In fact, the second working group meeting of Bioneca was organized in the capital, La Valletta, between the 6th and the 7th March 2008. I come from the Department of Metallurgy and Materials Engineering. It is a department that focuses mostly on material processing and surface engineering, and we have a number of industrial equipment. We also do a number of material characterization. We have a number of machines that can characterize materials and their surfaces, and also the bulk material characterization. And as you can see here, we had a number of short-term scientific missions that were held in Malta, precisely in January 2019 and April 2019. Here you can see Dr. Inge Massimo Rogante from the Rogante Engineering Office, myself and Dr. Rogante were working mostly on the surface engineering using a technique which is called PYRAC of nickel titanium shaped memory alloys. These materials have extensive use in cardiovascular stents, as you can see in the image here. And with his contacts, Dr. Rogante has conducted a number of characterization, mostly on small angle neutron scattering, ultra small angle neutron scattering, and also high resolution neutron diffraction. From the University of Malta, we're working mostly on the processing of these nickel titanium superalloys. Here we can see a number of different coupons with different surface treatments. Another project which personally I'm not involved in, however, Professor Eng Bertram Malia has started this project together with the Dane Technological Institute with Dr. Lars Plett Nielsen and Dr. Christian Rechendorf. The student, Janelle Arpam, is working mostly on implantable neural stimulating electrodes, which is also part of this cost action. Their project mostly focuses on the generation of porous titanium nitride, which has been used in implantable neural stimulating electrodes and which finds applications such as the restoration of sight and hearing, bladder control, pain modulation, and other applications. The main aim of this project deals mostly in order to solve the most important research gaps in the area. These are the mechanisms of the charge transfer from the porous electrodes, the potential for reduced impedance through surface engineering, and also to test the susceptibility to biofouling in vivo. I would like to thank you and looking forward to more Bioneca meetings in the near future.
Hi, from University of Genoa. Let me introduce our research center called BILONG, Biological Inspired Engineering, for Vascular Medicine and Longevity. Our teamwork covers a wide area of competencies including surgeons, biotechnologists, pharmacists, chemical engineers, and bioengineers. The core of our research is the study of modulation of inflammation and endothelial dysfunction, and how these processes are modulated by natural molecules such as polyphenols extracted by vegetables. Moreover, we develop other topics as vascular drug delivery, vascular tissue engineering, bioreactors, and fluidodynamic studies. Just to give you an idea, let me say that we demonstrated in vitro that one of the components of our extracts, apigenin, is capable to block the transcriptional proteins that promote endothelial dysfunction. We studied also resveratrol in vivo, inducing the development of an aortic aneurysm in rats. We demonstrated that it was reduced by treating them with the resveratrol. Coming to vascular drug delivery, when the atherosclerotic plaque is not calcified, it represents a possible target for drug delivery nanosystems. In our experience, we have synthesized different models of nanoparticles, such as liposome and polymeric ones. This is an explicative slide of the mechanism of the drug delivery of bioactive compounds on the atherosclerotic plaque. Nanoparticles are injected in the bloodstream and they reach the pathologic site where they release the drug.
Another field of research is represented by engineered biodegradable small diameter vascular prostheses able to interact with cells and to give rise to a new vessel. We used the electrospinning technique to fabricate vascular polymeric scaffolds. Our scaffolds are functionalized by loading bioactive compounds able to modulate the inflammatory process and to recruit cells. We also tested the biodegradable scaffolds in our bioreactor facilities before in vivo implantation.
I'm Professor Yannis Misirlis, retired but still active academically at the University of Patras in Greece. I was educated as chemical engineer, then biomedical engineer, working for the Artificial Heart Program in the US. From that time on, I have been trying to study and understand how life is working, how cells respond to different stimuli, what is health, what is homeodynamics. In ancient times, there was no specialization, but the concepts of balance, of interdependence, of harmonious coexistence guided the medical practice. That is interdisciplinarity. Here we see a marble plaque depicting Hippocrates at work. We see the symmetry of the four elements, the similarities of Greek and Chinese medical practice. I am a member of several cost actions, including Bioneca, where my interdisciplinary background and my dialectical methodology are catalyzing the interactions of excellent, yet of different specialty scientists, engineers, and medical doctors. In this sense, I am promoting the concept of mechanoepigenetics. As you can see here, the description of my concept, as well as a diagram of how it works. Another example is the design and construction of bioreactors to mimic the activity of living matter, cells, microbes, or their interactions, how they grow and divide under relevant mechanical signals. At the same time, you can see here that at certain conditions, cells divide because they sense the right mechanical signals. Finally, I wish to acknowledge my current active collaborators, Professor Stergios Logothetidis of the Department of Physics at the University of Thessaloniki in Greece, as well as Professor Aylin Sandemir from the Bioengineering Department at Ege University in Izmir, Turkey.
Probably each of us, at least once, was dreaming to become an astronaut and explore the space. We scientists are very lucky, because even if we are not going to space, we can make an experiment there. On Earth, we're working with stem cells. As you know, we all consist of cells. Thus, neurons are cells of our brain. We also have special immune cells which protect us from microbes. We have cells which are contracting and make our heart beating. We also have stem cells. This is a special type of cells which can generate any type of cells in our body. These cells can be collected and cultured in the laboratory. We can use different chemicals to guide their differentiation to any type of cells. However, physical forces also can affect the cell differentiation. Can you imagine how interesting to put the cells into space where there is no gravity at all? How microgravity will affect their differentiation? Working in the UK, I often travel between Stockholm and London. And to have non-stop experiment, I put my cells in the tube and kept them in the pocket. I noted that sometimes, several days, they've been out of thermostat, but when they placed in the stem cell medium, they continue to work very well. So I was thinking, why they cannot go to space? I came to the Swedish Space Corporation and said, my cells don't need any special treatment. Can they go with mother, it's a sounding rocket, to the space just to see if they can survive? Swedish Space Corporation prepares sounding rocket with several scientific experiments on board from the north, very north of Sweden. So our cells, they didn't need a special equipment. They were placed on the bottom of Mother 14, and on 26th of June, they went to space. Altogether, they've been out of laboratory for 2 weeks. And can you imagine when they came back and we placed them in stem cell medium, they survived. They dropped on the bottom, they looked very, very tired, but when we came in the morning, it was thousands of small neurospheres above them. So it was just explosion of stem cell proliferation. We collected these cells and compared them with Earth control cells, which also have been out of thermostat for 2 weeks. We also compared them with the cells which have been in thermostat, the normal cells. And to our surprise, the cells which have been in space, they possess absolutely new features. They have been expressing genes which protect them of different diseases, which make them better survival, more resistant to stress, and they've been better proliferating. Together with European Space Agency, we're now preparing for a new experiment where our stem cells will go together with beta cells which produce insulin, to help them produce insulin in space. This approach can develop to new diabetic treatment. We now can go inside and see how the hardware for this experiment is made by our colleagues from Swedish Space Corporation.
Neural crest stem cells in 3D bioprinted scaffolds have been one of the research topics on our last mission MESA 14 in 2019. The research team around Professor Elena Koslova from University of Uppsala and CellLink have found a couple of interesting features on the space-exposed cells. This leads now that the team is actually working on a new experiment to be flown on a sounding rocket again.
We are a team of passionate scientists and engineers pushing the limits of what's possible to pave the way for the future of medicine. With our 3D bioprinters and bioinks, we are pioneering the next generation of medical research. We work together with our collaborators in hundreds of labs across more than 60 countries to deliver the best quality and support. The magnitude of our team's knowledge and compassion and drive is enabling us to change the future of medicine. Through our collaboration with Professor Elena Koslova at the University of Uppsala, we are exploring how stem cells respond and adapt to the extreme physical conditions of spaceflight. We have prepared 3D bioprinted boundary cap neural crest stem cells using CellLink BioInk and the BioX printer, which were launched into space on board of a Master 14. The comparison of 3D bioprinted cells were subjected to altered gravity, mimicking space condition, or microgravity condition. We found that space cells proliferated and survived better compared to Earth controls.
The properties and the therapeutic benefits of ozone. Ozone, a two-atomic molecule made by oxygen, is a natural substance, very reactive with the biological structures present in the human body. For this enormous power, usually it is considered a poison and a toxic substance from which to keep well away. Instead, as it often results in pharmacology, it is a very powerful substance and in small doses becomes a powerful drug. Some experiments conducted with the Bioneca's activities in the last years have shown that a small amount of ozone is able to stimulate the activity of neural stem cells and their differentiation in harmony with the therapeutic properties of ozone at the clinical level. On this point of view, due to the activity of ozone to release ROS and LOPS substances reacting with the human blood, saliva, interstitial fluid, and other biological liquid substances in human body, the possible use of ozone in treatment of human diseases is numerous. In the last years, we have reached the witness that ozone derivative substances react with cells that are very important in the maintaining of homeostasis for important functions, as endothelial cells for normalizing hypoxia into the tissues, as leukocytes for normalizing inflammatory responses, as cells involved in cell senescence and cancer. All of us know very well the importance of oxygen and its allotropes to maintain the homeostasis. Organisms of higher animals relate to the stability of internal cell milieu with many daily rhythms, such as body temperature, secretion of hormones, and many others. Over time, the importance of organisms adapting to various stimuli has become increasingly evident, together with the importance of the response of each organism in relation to its own specificity. Mild, acute, and targeted oxidative stress is well described as oxidative eustress, and it elicits adaptive responses in cells. On the other hand, oxidative distress is the result of chronic, uncontrolled overproduction of noxious metabolisms that can result in tissue damage. Ozone therapy, with the characteristics and specific multiple methods of administrations, fit well in this context. Just to summarize, any time we have to fight against the diseases where the etiopathogenesis is referable to infections, inflammation, or regenerative disorders, especially when hypoxia is present, the ozone-oxygen mixture is a valid help in healing treatment. During this awful year, 2020, COVID-19 changed our concept of social life and a lot of our behaviors. Many people died for a disease that started as a respiratory infection, changing after weeks or days for some of this in multi-system failure of organs due to the impaired function of the endothelial cells. The ozone therapy, meaning the possibility to let the ozone derivatives, ROS and LOPS, to achieve the microcirculation vasculature and protect the cells from that sudden change, showed to avoid this passage from SARS to COVID and contributed to save many lives. To achieve these results, we have made many treatments in private and public structures, and we obtained good and numerous early healings. Treatment with ozone is safe, with no side effects, and is very cheap. It lasts at least 40 minutes, and the procedure provides that to take an amount of blood of 100 to 180 milliliters, but put in a certified plastic bag, gently mix the blood with a mixture of oxygen and ozone at a low and increasing concentration of 10 to 30 micrograms of ozone in 1 milliliter of oxygen. Then, after 4-8 minutes, let the blood reinfuse in the same vein where it was drawn. This has to be repeated for 3-4 times a day, and even if patients were treated once a day for 3-6 days, good results were achieved. In conclusion, we may define the ozone a wonder drug for a medicine of the future on a human scale.
Hello, my name is Dinko Mitricic. I'm a professor of medicine at the University of Zagreb School of Medicine, and I will present you briefly our work. University of Zagreb is approximately 500 years old, and among many expertise we offer to worldwide scientific community, one of the probably most famous is field of neuroscience. Just I will give you one example. We have the third largest brain bank in the world. So we have huge collection of human brains in different various embryonic developmental stages, as well as adult brains, healthy and coming from different diseases, which are source of numerous publications we published together with our international partners. Laboratory for Stem Cells, which is guided by myself, has expertise in several types of stem cells. Thus, we offer expertise in neural stem cells, mostly obtained from mouse animal models, but also we have experience in human stem cells, mesenchymal stem cells, but also some specific population of stem cells like those ones coming from oral mucosa. Our major expertise is in defining these stem cell populations, in analyzing their profile of their secretome, and also our expertise is focused on transplantation of these stem cells. So we have many publications in the field of transplantation of stem cells in animal models, and then analysis what occurs with animal after transplantation, what happens with tissue after transplantation, but as well what happens with stem cells after transplantation. To perform this task, we built a completely new laboratory that was built in 2015-16 approximately. It is a laboratory which consists of numerous elements which are highly technologically developed. So we have a laboratory for stem cells with high-tech and high-end technology equipment there. We have a special room for operations of animals. We have an animal facility. We have a room for bioluminescence. So we have equipment for bioluminescence as well as magnet resonance imaging, Bruker 7 Tesla. So combination of these facilities all together, from stem cells, animals, over imaging facilities, both histology, bioluminescence, and MRI, is a unique combination in this part of Europe. Actually, there are only few places in Europe which has this combination of expertise at one place. So this is how it looks like. We have Bruker 7 Tesla, bioluminescence, and numerous other instruments. As I said, we isolate stem cells both from mouse embryos or adult mice, but also from humans. So we have several ongoing projects. For example, some of them are based on cells from human oral mucosa. Also, we are developing technology for three-dimensional cell cultures like organoids. We already have our first publication published in this field, where we can develop, for example, small pieces of human brain in dish, which can give you a lot of advantage over 2D cell cultures. For cell analysis, we have several very modern instruments. One of them is automatic station for following of cell differentiation. In this video, you see how we follow cells while they migrate in real time. So we can follow real-time migration and differentiation of neurons, for example. These cells are then, for example, transplanted in animal models. So in this video, you see mice which are suffering from stroke. They have brain ischemia, and mice is circling in one direction as a proof that it suffered brain ischemia. Then, in these animal models, we can transplant stem cells, various types of stem cells, and various protocols on transplantation of stem cells. And then these animals can be analyzed using our high-end imaging facilities like bioluminescence or magnet resonance. And then we can perform all type of histological analysis, immunohistochemistry, transplantation analysis, migration studies, biochemical studies, proteomic studies. And what we are really developing fast in the last few years, there are some automatic analysis of large number of cells, both in culture and from, for example, histological sections of human brain. So we are developing special protocols which allow us to systematically analyze elements of cells, sometimes subcellular elements, and then to perform some mathematical analysis, which finally gives us results about profile of these cells, for example, before and after transplantation. So this is the end of my presentation. Once again, we are Laboratory for Stem Cells, University of Zagreb, School of Medicine.
Check My Brain is a simple yet most innovative medical test which helps millions of people worldwide. The chances of being affected by Alzheimer's or other brain diseases is more than 50%. One major reason as why we are currently unsuccessful with the treatment of brain diseases is the lack of simple tests for detecting disease in its early phase. To check other organs such as your breast or stomach is rather simple. The brain, however, is almost impossible to access since it is positioned underneath the skull, hidden behind the blood-brain barrier. This means that blood tests or scans cannot detect brain disease in its early stage. Check My Brain is a breakthrough in medical diagnostics. It is a simple yet very efficient test based on the most recent discoveries in medical research. How does it work? You need to simply sacrifice only 20 hairs and 20 seconds of your time. Pluck 20 hairs from your head and send them to us. By using stem cell technology, we will transform your hair cells into your own brain cells. They will allow us to detect possible earliest signs of developing brain disease. If we were to observe any abnormalities, we will then be advised to perform more detailed testings in the clinic in order to confirm or deny our findings. And don't forget, if your disease is detected in the early stages, you will be treated immediately in order to postpone the progression.
My name is Leonora Buzarska. I'm director of Mosakowski Medical Research Institute, Polish Academy of Sciences in Warsaw, Poland. This is the Institute of Experimental and Clinical Medicine. Thus, our overall goal is implementation of basic biomedical science to the clinic. Two departments from our institute are involved in the Bioneca cost action, namely Department of Stem Cell Bioengineering, headed by myself, and Translational Platform for Regenerative Medicine, headed by Professor Anna Sarnowska. The following film presents our research profile and experimental models.
Hello, my name is Rodolfo Martinez. I am IP of a group working in neurology and stem cell, and we are members of Bioneca project. We are working in Spain, in Alicante, in the Institute of Neuroscience, and also in Hospital Virgen de la Risaca in Murcia. Our interest is to discover cellular therapies for neurodegenerative disease. We are working in ALS. We know that ALS is a primary degeneration of the motor system, both in the neurons of primary motor cortex and the neurons in the spinal cord. And this disease has a very bad outlook because most of the patients die between 3 and 5 years after diagnostic. So the motor neurons that are these cells that disappear progressively in the disease, and we have studied if adult bone marrow stem cells are able to protect those cells from degeneration. We are using both hematopoietic and mesenchymal stem cell from the bone marrow, and we are using those cells as biodrugs in cell therapy experiment. So in animal model, we have demonstrated that the grafted bone marrow stem cells are able to survive in the spinal cord. And in addition of that, they are localized around the motor neurons that are labeled in red. So grafted cells are labeled in green, motor neurons are labeled in red. So the trophic effect of grafted cells are just acting close to the target cell that are the motor neuron. We have demonstrated that this trophic activity of bone marrow stem cell acting over motor neuron occurs in vivo and in vitro. So those result has determined the development of clinical trial phase 1 and phase 2 grafting autologous bone marrow stem cell in ALS patients with different kind of controls, and we have demonstrated that the grafted cells also are localized around the survival motor neurons in the grafted segment of patient spinal cord. And also in vitro we have demonstrated this neurotrophic activity of bone marrow stem cells. In addition of this histological effect, we have determined that in magnetic resonance studies the neuronal activity of primary motor cortex was improved in the grafted patient and in the grafted side in relation with the contralateral side of the non-grafted patient. So suggesting that there is a retrograde activity, neurotrophic activity in this kind of therapeutic approach. Therefore, we have started a new experiment, just grafting cells in the muscles, just looking for retrograde activity into the spinal cord. And the animal model demonstrated this retrograde activity from the muscles, not only the survival of motor neurons in the spinal cord, but also the maintaining of the shape of the neuromuscular endings. So we have promoted and developed phase 1 clinical trial, just grafting autologous bone marrow stem cell in one side of anterior tibialis muscle and the control injection in the contralateral side, double-blind randomized clinical trial. And this trial, phase 1, showed that this graft was safe in the patient, so there is something that can be done without any problem. And in addition of that, an improvement of activity in the grafted muscle in relation with the control contralateral side. So suggesting that in addition of the safe procedure. We could also improve the activity in the muscle. So now we are just starting and phase 2 clinical trial with intramuscular injection of bone marrow stem cells. We are a basic and clinical team working in the 2 hospitals and in the university, just looking for the activity of this cell therapy and the potential new therapies for ALS. Thank you very much.
Hi, my name is Pablo Andrews, and I am the head of the Center for Laser Microscopy of the Faculty of Biology, University of Belgrade. We are devoted to develop NemoChip, a neurodiagnostic lab on a chip that is disruptive for the market of in vitro diagnostics. Our team is made of in-house biologists and biophysicists and external collaborators, photophysicists and technical engineers. Early and precise diagnostics for neurodegenerative diseases is generally lacking. When a doctor sees a change in a brain scan, it's too late. And we often do not have efficient therapy that can alleviate that burden. Many neurodegenerative diseases are characterized by neuroinflammation. It's a complex picture, however, crucial for neuroinflammation are the antibodies, or immunoglobulins, especially autoantibodies. Our research focuses on neuroinflammation in amyotrophic lateral sclerosis and the role of immunoglobulins in this particular motor neuron disease. ALS is a complex neurodegenerative disease. Diagnostics of ALS is challenging. In only 10% of the cases, we know the genes that are responsible for the disease. However, the life expectancy is still only 2 to 5 years. Our expertise is based on experimental preclinical work and dynamical recordings from fluorescently labeled neuronal cells. All our expertise in the lab we put on a single lab on a chip so that we can analyze small samples of patient sera or immunoglobulins in a standalone device that we call the NemoChip. This way, we would offer better personalized diagnostics that would hopefully lead to better patient stratification. The overall goal is to develop the procedure that can analyze multiple parameters from time series recordings that can help clinicians make better decisions. This way, we believe that we would lead to personalized treatment, not just for ALS, but for various brain disorders with neuroinflammatory manifestations. Thank you.
Hello, my name is Alberto Brevin. I'm senior scientist at the biomedical mainline of the European Synchrotron Radiation Facility in Grenoble, France. Bioneca, I'm focused on high-resolution multi-scale X-ray imaging in collaboration with institutes, laboratories and hospitals all around Europe. Synchrotron radiation is generated inside large-scale facilities, where electrons, accelerated to the speed of the light, circulate inside the storage ring. When electrons enter inside powerful magnetic fields, they generate the most intense X-ray beam on Earth. This beam is then transported inside the nearby laboratories to perform X-ray imaging. Due to the high intensity, computed tomography imaging of the entire rat brain can be achieved within a few minutes. Synchrotron radiation is also highly coherent, and this permits to obtain high-quality images like these ones using the phase contrast technique. The neurons can be visualized in 3 dimensions in a mouse's spinal cord. This is an extremely powerful method when studying the evolution of neurodegenerative disease. Additionally, also the interface between vascular and neuronal systems can be highlighted using pixel sizes of 0.75 microns. Without using any contrast agent, we can visualize and navigate inside the vascular and microvascular system in a rat brain. Using even smaller pixel sizes, down to 0.3 microns, we can focus on the somatosensory cortex and characterize the vasculature, for instance measuring its density or determining the number of branches. And more importantly, this analysis can be performed to post-mortem also in human bodies, examining the spinal cord white and gray matters simultaneously with the microvasculature. And this can be performed not only on a small portion of the spine, but navigate in 3 dimensions inside the full human spinal cord.
Good morning to everybody. I'm Silvia Di Angelantonio and I'm going to show you some of our recent results of the identification of ocular biomarkers for the early diagnosis of Alzheimer's disease. Alzheimer's is the most common cause of dementia and general term for memory loss and other cognitive abilities serious enough to interfere with the everyday life. And actually, Alzheimer's has not a cure, and the only possibility is to stop the neurodegeneration at its initial phase. Indeed, what has been demonstrated in the brain of Alzheimer's disease patients is that neuronal loss probably caused by protein aggregation and neuroinflammation appears long before the first sign of dementia. So the idea is to find a way to early diagnose Alzheimer's disease in order to possibly treat the patients. And it has been recently shown that visual and ocular deficits and also other histological hallmarks as protein aggregates can be found in the retina of Alzheimer's disease patients. So the actual challenge is to define new biomarkers and non-invasive technologies to measure the neuropathological changes in vivo at pre-symptomatic stage to allow differential diagnosis and to maybe, possibly, introduce a therapeutic intervention into patients. And we use to detect these possible hallmarks a model system that is the retina of Alzheimer's disease patient from autoptic tissue and age-matched controls. And we look at first at the presence of protein aggregates of beta amyloids in the extracellular space and tau tangles in the intracellular compartments. And we found that in Alzheimer's retina, there is a stronger accumulation of beta amyloids in the extracellular space, tau tangle in the granular cell layer in the intracellular compartments, and possibly due to this protein aggregation, also an increase in neurodegeneration of retinal ganglion cells that are the cells that form the optic nerve. So we moved also to look if, as in the case of the brain, neuroinflammation can occur also in the retina of Alzheimer's disease patient. And what we found is an increase in microglia cells number and also an acquisition of a detrimental Alzheimer-like phenotype of microglia, and also an increase in astrocyte and an acquisition of neurotoxic phenotype of astrocytes. So to conclude, we can imagine that retinal biomarkers such as protein aggregates, neurodegeneration, and the activation of microglia and astrocytes together with a development of the detection method may become a strong tool to early diagnosis of Alzheimer's disease and to monitor Alzheimer's disease progression. And we hope that this will pave the road for a new cure, a new treatment for Alzheimer's disease. And I thank you, all of you, for your attention.
In continuation of more than 50 years long tradition of electron microscopy at the University of Science and Technology, International Center of Electron Microscopy for Material Science has developed its infrastructure. The purchase of the microscope Titan G2 6300 with ChemiStem technology was supported by the structural funds of the European Union. At present, the International Center of Electron Microscopy for Material Science played the role of TM training site for Central and Eastern Europe and collaborates with 40 research institutions from 18 countries. Research activities of the International Center of Electron Microscopy includes basic and applied research related to the problems of electron microscopy in material science, physics, chemistry, electronics, energy and medicine.
My interest concerns the characterization of advanced material by large-scale European facility, like intense neutron source synchrotron radiation. However, in order to obtain very high spatial resolution till the level of nanometer, is necessary to come in Krakow and to use this famous microscope Titan.
This is one of the most advanced transmission electron microscopes in the world. Its high resolving power of 70 picometers allows us to observe microstructure and nanostructure, as well as measure chemical composition of the materials even at atomic level. Thanks to this, we are able to say which atom belongs to which element. Typical sample for Titan have dimension of 10 by 7 micrometer. If we will magnify it by 10 million times, it will undertake the size of the football field. At such extremely high magnifications, we are observing area of 10 by 10 nanometers, what gives the equivalent of 10 by 10 centimeters square on the football field. In certain conditions like high stress or elevated temperature, material properties depends on its microstructure, which is a result of the certain chemical composition, heat treatment, and processing of the material. Thanks to our equipment, we can investigate that structure, link it with the material properties, and to consider how the material can be improved to make it suitable for the certain application, like energy systems, aeronautics, biomaterials, or automotive industry.